Hi everyone, and welcome to a brief walkthrough of the Preparing Solutions and Dilutions lab that you have as a take-home. So in this lab, you have in your kit your digital scale, you have a little packet of blue Hawaiian punch, and you have 5 plastic shooter glasses. So before you can even do this lab, you have to go through the calculations in order to do it correctly. So what you are doing essentially is you are going to be preparing a stock solution, so a more concentrated solution of this Hawaiian punch, and then you will be diluting it, doing a couple of dilutions. You'll put all of these different concentrations in each of those shooter glasses, and you will measure the concentration and confirm that you did your dilutions. You made your stock solution and did your dilutions correctly using Beer's law, which is a law that allows us to basically detect the amount of light that passes through a substance and see the decreasing color, measure the decrease in the color, essentially. All right, so the first problem, problem 1, asks you to calculate how many grams of the Hawaiian punchide you need to add to water in order to make 500 milliliters of solution that has a concentration of 0.08 molar. And so you can do this using the equation for molarity. Molarity, of course, is moles of solute over liters of solution. So in this problem, we know what the volume of the solution is, 500 milliliters, so you'll need to convert that to liters. And we know the molarity, the desired molarity is 0.08 molar. All right, so the variable that we will solve for are moles. And then from moles, you can convert to grams using the molar mass. All right, so that's the first step. Then you will actually do that. You will measure out that many grams of Hawaiian punch, add it to your beaker, fill with water to the 500 milliliter mark, and then you will put 60 milliliters of that concentrated solution into shooter glass A. So this is your most concentrated solution. Then you're going for problem 2. You will need to calculate how much of this solution you need to add to each of these shooter glasses in order to make 60 milliliters total volume of each of these concentrations. So you'll be doing 3 dilution problems in part 2, question 2. And remember the equation for solving dilution problems, C1V1 equals C2V2. Initial concentration times initial volume equals final concentration times final volume. The initial concentration in these problems is going to be our stock solution concentration, and our final volume or final concentration for each of these will be the concentration indicated, and the final volume for all of them will be 60 milliliters. We want all of these to have the same volume. Okay, so what you will be solving for is volume 1, the volume of this solution that you need to put in here in order to bring it up to 60 milliliters with water. So this table is just a place for you to sort of organize those results so that when you are preparing these different shooter glasses, when you're preparing those different solutions, you can just follow those amounts on the table. So you'll be adding some amount of this solution and some amount of water. And those 2 amounts need to add up to 60 milliliters total volume. All right, so that's as simple as it is. That's all you're doing. And then you will line up those shooter glasses with a piece of white paper behind them and download this app on your phone that allows you to basically take detailed color data, to take a picture and get detailed color data from that picture. And then you will record that data on this slide here. It breaks down the colors into different pixels, red, green, blue. So for each of the shooter glasses, you'll record the data that it gives you, and then you will input that data, actually just the R values, just the red values. You will input into the Excel spreadsheet that's in the lab posted on Blackboard. So you go to Blackboard, Labs, Solutions and Dilutions Lab, you'll see an Excel attachment. Open it up and just type in these 5 numbers and it'll auto-populate a graph for you. Take a picture of that graph, maybe a screenshot or a snip, or use your phone and take a picture of your screen, however you do it, so that you can include that in your files that you upload as part of your results from this lab. All right, so that hopefully is clear. If you're having trouble with the calculations, of course, let me know, email me. That's all.